And welcome everybody to our fourth webinar in our series for summer PD. This webinar is entitled Responding to the Mystery Illness. So along with talking about ABE specific assets, we're going to be focusing specifically on responding to a mystery illness class. And we're going to take you through that and show you how you can use it and modify it. We're also going to talk about some additional assets that are available on LabXchange that are not part of the class that you may want to include if you decide to use this with your students. First of all, we're going to go ahead and just talk about, we're going to lay out the evening for us. Well, evening if you're on the East Coast. We're going to go over some brief introductions, a general quick overview of LabXchange. We're not going to get too in depth with this overview of LabXchange, as I've mentioned in prior webinars. If you need a refresher or you're just interested in some of the nuances of LabXchange, Jessica Silverman is offering 1-hour webinars through LabXchange, and she has those recorded as well. So we can get you those recordings if you'd like to watch that. Or I believe that there's still a couple of live sessions. I can look and put the link up in the chat for that. In terms of our focus for this evening, we're going to focus on just ABE specific assets and how you can use LabXchange, focusing on ABE for that. We're going to take a look at the responding to a mystery illness practice and some questions and answers. So the important thing about the webinar today, this evening, whatever time zone you're in, is that it's for you. So if you have questions, now is the time to ask. And if you're not speaking, if you could just mute yourself, that'd be great. If you have any questions, please ask at any time, raise your hand. Although I'm terrible at seeing the hands, I'm better if you do the chat, or you can just, interrupt and say, excuse me, and unmute yourself and talk. We're a small group this evening, so you're welcome to do that at any time. So if you are not speaking or you don't have anything to say, if you could just mute yourself to just take away from the background noise, that would be great. We are going to be using these webinar recordings in our blended PDI course. If you are registered for this webinar, you will be receiving a link to fill out our release form that requires your signature and then to be emailed back. So that will be coming from the program office, within 24 hours of receiving the links for the webinar. So let's get started if nobody has any questions so far. Introductions, I'm Jen Uroff. I am the online learning specialist for the ABE program office. Prior to my time at EDC, I've been at EDC for about 8 years. Prior to that, I was an elementary teacher where I taught everything as an elementary teacher. So at EDC, I focused mostly on online learning. I've developed curriculum for lots of different projects and have worked in lots of different facets. And also with me today, Kristen, I'll save you the introduction. Kristen Bjork is my colleague. She is the ABE Content Development Director at ABE as well for the program office. She's been at EDC for 32 years, I think, Kristen, right? She will jump on later. And so she has a significant amount of experience and expertise in this area. And I'm going to pass it over to Deb, and she's going to take us right into that introduction to LabXchange.
Hi everybody, my name is Deb O'Reilly and I am currently a high school biotechnology teacher at Essex North Shore Agricultural and Technical High School. And I have been with LabXchange since it was just a thought and been working on developing curriculum, and this year I've taken on a new role to kind of work with both EDC and LabXchange and Jen. So welcome everybody to our webinar. We're going to talk a little bit today about just some general things. So if you've attended these before, you're going to see some of the same introduction. LabXchange has a lot of different and diverse pieces to its platform. And Jen, did you want me to share my screen to go over those specifically, or do you want to keep the PowerPoint up?
I think it would be great if you could walk us through on LabXchange, if that's okay.
Yeah.
Show and tell. Okay, so I'm going to stop sharing so you can take over.
Okay. Oops. Okay. So when you first click onto LabXchange, this is the main page here, and this little browse menu up here is where you're going to start most of the time. You can, be on the platform without ever like joining or, creating a login, and you can search through the library for different assets. The assets would be like, you know, video, scrollables, simulations, text, interviews, things like that. Or you can join LabXchange and create an educator platform. You on this dashboard, you can sign in either as an educator if you are the one running the course, or you can sign in as a learner if you are the student in the course. What's nice about that is as a teacher, you can have your educator dashboard and be running your classes, but you can also run PD or be part of professional development and be the learner and have someone else be the educator for that particular time, which is the case today, right? So we're all learning from Jen and about the new curriculum up on ABE. So one of the things that you're going to want to do in your educator dashboard, and you guys all did this, is join a class. But I'm going to go through the different types of assets that are available up on LabXchange. So if I go back to browse, I'm going to go into the library here. And there's different ways you can search. And, unfortunately, I don't think the search mechanism of LabXchange is perfect yet. I know they're working on it. So it can be a little bit complicated. So I recommend narrowing what you're looking for, either by content type or knowledge or subject area. So don't just search the entire library for something. For example, if I know that I'm looking for something in biological sciences, I don't need chemistry, health, I'm not looking for online learning, I'm going to narrow it there. And if I want something specifically from content type, maybe I don't want a text, maybe it's a video and a pathway maybe, or a simulation. Those are the things I want to narrow down. And then say I want to do something on sickle cell. Oops, I spelled that wrong. I can put it up in my browse window and search, and that's going to help narrow down the information you're going to get back to be more specific to your search. It's kind of like if you search on Google and it's too broad and you get just an overwhelming amount of information, you really want to narrow it down a little bit, and the better information you can give. So with that, I want to talk a little bit about the types of assets that we have up on the platform on LabXchange. There's text assets. So here's an example of a text asset here. You can see that, sorry for my slow internet today. You can see it's just a text with images. And what's nice about the text on LabXchange, it's not going to overwhelm a student by the length of the text asset. It's not going to be pages and pages. You can look at images. You can see there's a bunch of text. And I've decided, all right, I don't necessarily want a text image. I want to look at just pathways. And then I'll see a pathway. Now, what's a pathway? If you're used to teaching units or a lesson plan that might involve like a series of lectures or events, labs, or texts that you're going to do, you can think of a pathway really as being like a large lesson plan that's going to include lots of things. So I'm going to go back up, clear all of my browsing and go back to the beginning so that I can kind of look at this. So what's nice about LabXchange is it has this other piece of the process called a cluster. I'm going to click on the clusters. Right now there's only 5 clusters, but that is one of the contents that's being worked on. They're working on a PCR cluster, working on some higher level clusters for grad level students. But so the one that you guys want to, as ABE teachers, and the one I use all the time as an ABE teacher as well, is this foundational concepts and techniques in biotechnology. This cluster here is where you're going to find most of the ABE curriculum that, or at least that supports the ABE curriculum currently online. And within clusters, you have this button here that's called tours. And what that does is that helps you to narrow down, because you can see there's a lot of hexagons in this cluster. So it's got a lot of information here. It could help you narrow down and kind of match up to more of the ABE curriculum a little bit at a time. So for example, if you wanted to introduce your students to pipetting or gel electrophoresis, and these names are going to change to support the ABE naming. So they'll have Lab 1, but they'll also have the other, Jen, do you remember what the other names are?
So Lab 1 is tools of the trade.
Okay.
Lab 2, I don't know off the top of my head. I just
That's
know
fine.
Lab 1.
Just
Okay.
you're going to eventually have both tags, both name
Correct.
tags, so
Yep.
that you'll, whichever way you're familiar at looking at these. So if you look at Lab 1 and I click on it, it highlights those assets, those pathways that are coordinated with those, with the ABE labs. So for example, teaching pipetting, you go into this pathway and it's going to provide you with lots of different things about this pathway. And if I go back to my library catalog, this is the one thing that is a little bit annoying. Is that you kind of have to go back in it the way you just did. You had to go here to your cluster. It doesn't have like a back button per se. And that is also something they're working on changing. So, if you wanted to do, say you just wanted to do Lab 6 and look at protein purification. So you're going to do your genetic engineering, the restriction digest. Here, you're looking at separating your recombinant protein. And one of the things that's great about this is once you have an already created pathway on LabXchange where they've put some things together. As a teacher, I always like to take what somebody's offering and then massage it so that it looks like I created it, or it fits more to what I'm doing specifically. So you have this ability to clone a pathway. So if you look at these 3 little buttons up here, those are important because that's going to allow you to clone the pathway, which means you can make a copy of it, and now you can edit the pathway to be something that you want it to be. And this button's important too. So if you have a glitch or something's not working, you can click on that and it will allow you to report it right to the programming office. So if I clone this pathway, what that's going to allow me to do is then edit this pathway. I can rename it to be, more personalized. Once this pops up, I can name this Miss O'Reilly's Biotech. Jen, if I'm reached on time, just let me know. Tools and techniques, and then maybe I don't want to show 2 videos in a row and I don't really think my students are ready to look at protein packing. I can just delete this out of the pathway and remove it, and now I've kind of done that. If I want to add content, maybe I've created a Google Doc that I want to add into the pathway so that I can check students' understanding of the video. I can add new content and I can create a text or a document, however you want to do it. And it's going to tell you, do you want to publish this privately? Yes, because it's really just for my classroom. And then I can just put the link into the Google Doc here and just enter the URL, the title, click OK. And then and upload it here, or I can give the students notes about what they're going to be doing here as far as, that particular worksheet goes. So it does allow you to kind of personalize LabXchange a little bit. So I'm going to go back to the dashboard here. So the other thing you can do when you're searching for information is favorite something. And I recommend that you do this a lot because there's so much. I mean, if we go back to the library, and there's so much information up on LabXchange now, and it's just growing by the day. If you look at here and at all these assets, it's a lot to search through. So once I've found things that I like, for example, I really like this coronavirus pathway. I'm just going to click on this little star right here, and that's favoriting it. So if I now go back to my dashboard into my educator dashboard and look under favorites, you're going to see that coronavirus pathway, I should probably clean this up when I'm doing this, is in here. I think I just passed it. I just saw it at the other side, but it will be in, there it is. It's in here right here, and it's in the favorites. And what that allows you to do is then when you go to a classroom, if you want to assign information to a class, you can add content into your class and take it. I apologize for the slowness of my internet today. It's been awful all day. You can put stuff into your classroom that's just content that you're looking to assign, and you can either assign it to your class by coming down here and clicking on the plus button, and that will now be in material that's assigned. And then if you're done with something, you can come here and you unpost it. And one of the other things that we haven't really talked about that I really like as an educator is I can come up here and I can see this learner view right here. I can click this on, and now I see what my students see. So they don't see all that other stuff that's up as my content. They only see what I posted, which is really nice. And I can go back and forth between the two views. So you get an idea of what they are seeing as well as what you have available. And I just want to quickly go back to this main page here, which is where you want to start if you really want to learn about LabXchange. There's all kinds of assets up here as well that have been developed. Here's a pathway on how to teach with LabXchange, a cluster about remote learning with LabXchange. If you need help, how you can find it. Then there's like how to make a pathway videos, all kinds of information. And then down here in the blog, if you're looking for any of the LabXchange tutorials or webinars that are still available, that's where you're going to find the information there. And if it comes up, so you can kind of click on any of these to see what workshops are still available. They'll be all scheduled. Okay, so I think I am all done. I think I covered everything. Did I miss anything, Jen?
I don't think so, but now's a good time to stop and ask if anybody has any questions related to what Deb went over, or if you're having trouble with anything or any clarification. We're good? Okay. I put some notes in the chat just about setting up an account, but for people who have been through this before, they know that. So next we're going to go ahead and we're going to move forward in to Kristen. So Kristen, are you ready to talk about mystery illness?
I am ready. Okay. I am ready to talk about mystery illness. Hi, everybody. So everybody, I'm assuming, has now joined the class, and you don't have to go to it right now. I will walk you through some of it, and then you can play with it on your own, and then we could answer any questions that you have, or I'm happy to answer any of those questions later. So what we did is similar to what you as a teacher would do with LabXchange, which is to take individual assets and mix them up and create a class. And in this class, we have a number of pathways that make up the class. And that's exactly what you would do as a teacher if you wanted to make your own. What we're trying to provide for ABE teachers is individual assets that are related to the ABE curriculum. So there are a number of the assets that are from the labs, such as Did You Knows, or particular sequences from the actual lab text, or some related images that you can just use individually. So some of the plasmid images are in just as plain images, and you could use those. So those are all in. And then there's the ABE specific curriculum cluster, which Deb showed you, the foundational concepts. And then there are tours, again, Deb showed you, where you can click on the various lab names, and it will show you what the related cluster cells are. And then we have ABE online classes, and this one is responding to a mystery illness, and we're also developing more, including online ABE labs that can be used to accompany in-person work with the labs, so either as remediation or practice, and or can be used in times like these for remote teaching. So responding to the mystery illness was made, surprisingly, for the COVID pandemic, and we wanted to build a set of resources that was related to the pandemic, that would be interesting to students, and that would also teach them some biotech or bioinformatics content. So here are the learning goals. I'm not going to read them to you. You can take a look at those, and then I can show you just a little snippet of the class, and you can get a sense of what we did within the class. Let's see if I can actually share my screen. Share. So here's the class. I've already gone into it, and this is what you would see when you actually entered the class. And here are the individual pathways that make up the class, and there are 6 of them. The first one is the story setting part of the class, and in it, students are introduced. Of course, here's one of the tricky things about LabXchange. If you leave off in the middle of a pathway, it will bring you back to that spot, which is lovely if that's where you want it to be, but I didn't want to be there, so I have to go back to the first page. The first page is just the setup page here. The kids are introduced to the beginning of the pandemic, and they're given their task. And we've included some assets from outside of LabXchange, which are in boxes here. This is a news report from China. And then in this call out box here, the students are given their call to action. So they're part of the WHO team. Their task is to help the monitoring team determine what organism is causing the mystery illness, and they try to halt its spread and begin the work of developing a vaccine. And then, so they've watched this video, they've read this text here, and then they answer a few questions just to check for understanding. And these are checked by the LabXchange machine. And so if they get them right, it will tell them. They can keep doing it over and over again. It's not, we did these as just simple checks for understanding, but if you assign the class, you will be able to see students' progress and see what they got right and what they got wrong. Again, I'm not going to do all of these, but we put in all these checks for understanding. And this is a different kind of check for understanding. This is a short answer question or an open text question. So they can answer, this is monitoring the temperature, but they can answer temperature, fever, one other thing, I can't remember what it is, and it will mark them as getting it right. And then at the end of each of our sections of the mystery and illness class are discussion questions. And what we've asked them to do is just prepare themselves to discuss these questions with their class. We have a Google Doc that has suggested answers to these questions, as well as the answers to the multiple choice and the short answer questions. And we can give you a link to that if you just let us know. I've got to go back to the class. So again, when you go back, you're going back to the first section of the class, which is this pathway, a mystery illness strikes. And then you need to go back to the class here. So in the second section, their job is to identify the cause. And so they get a little taste of how you would get a sample. And we found a nice video of somebody doing bronchoalveolar lavage, which is somewhat gross. So we put a note that said that they didn't have to watch it. They could read about it on Wikipedia. And then you can see how a test is done in this video. And then we used a LabXchange asset here where the scientists from the African Center of Excellence for Genomics and Infectious Diseases, which has a great acronym that I don't know how I would say it, but I like it. And then they could watch this video, gives them a lot of information about how PCR is done. And then their call to action is, as part of the WHO team, their task is to help identify the organism. And they learn to use BLAST as a bioinformatics tool. We've built in a screencast here that shows how BLAST is used. I'm not going to play it all, but at least what it does is it sets them up to be ready to use the actual BLAST tool, and that's what we ask them to do. So they take this sequence here, and this is the known illness. So this is set up to be a patient who has a known illness, a known pneumonia, and they use BLAST to actually identify this sequence here. And it shows them how to do it in the previous tutorial, and they would go out of this part of the pathway, use BLAST, and then come back in and continue. Then they're given an assessment that says which one of these was it. I think it's influenza A. Now I can't remember. Oh, I got it right. And then they are asked what type of organism the virus might have come from. And I'll show you here. This is the unknown sequence here, and we had to use a graphic for the unknown sequence because if you put in the actual sequence from coronavirus, it will tell you it's coronavirus and this current SARS-CoV-2, and we wanted them to not know that it was SARS-CoV-2 right away because obviously these WHO scientists back in December wouldn't have had any idea that it was SARS-CoV-2. So we wanted to keep it as realistic as possible. So after they've identified, and they identify here that it came from bats, and they submit their answer, then they're given more discussion questions, and they go back to the class. Then they learn more about the cause. So this is basically, they're learning about viruses here. And again, it went to the spot that I had left off, so I apologize. Deb, do you know if there's any way to get it to just start at the beginning each time? Oh, we lost her.
Sorry, I'm on mute.
No, yeah.
I just explained it to you. You didn't hear?
Oh, darn it.
Sorry, you missed it. No, yeah, so the reason it doesn't go back is because you monitor student progress in an asset, right? So like if you've given them a pathway, it'll tell you they've completed 20% or 30%. Right. So if it went back, it would reset on the student.
Got it.
Case, they would not be happy with us, right? Like, so it's kind of that, oh God, like which way do you go with it? But as a teacher, you kind of want to know how far along they've gotten. So I'd rather that it be a little bit cranky about you going back.
Yeah, I think it's just important to tell students when they're using this that they are going back to where they left off so they don't get confused, because I don't always notice myself these little bars at the top, and so then I go in, I don't know where I am, and so just giving them that heads up before you send
Right.
them off to do this is important. So in this pathway, they are taken to yet another spot in the timeline. It's in February, and they know that this pathogen is a virus, and they need to learn about viruses. So we give them a video about viruses, and then LabXchange has some really nice assets from OpenStax where you can read about viruses and learn about the viral replication cycle and the life of a coronavirus. And then they can learn about why bats don't seem to get sick from the viruses they carry. And it's another reading for them. And then about zoonosis. So these are all just getting them familiar with viruses and how they work. And then another quiz. And again, these are, we try to tell kids these are checks for understanding for them, we're not trying to assess them, but of course you do know whether they got the answers right or not. And then again, the discussion questions. And then what should we do right now is simply asking students to think about, what do we do now that we have this disease and we've identified it? What are we going to do to help people? And again, it went back to where I was. So I have to start over here. It's in February. People are getting infected everywhere. Your team at WHO recommends communicating with the public so they can help stop the spread of the disease, and then look at these solutions and think about how you might stop the disease from spreading. So they learn about biohazards and public health or epidemiology from OpenStax here, and they learn about epidemiologists' language again from OpenStax. Then how infectious diseases are tracked. So this is all really important information for students who are learning about viruses and how they can be mitigated. And then here are two sets of communications that people have used: this WHO's video about how you can protect yourself, and a poster from California Department of Public Health. Then again, the knowledge check here and your discussion questions. And I think in an in-school activity or even an out-of-school activity, you could have students design their own public information campaign and submit it and see, you know, vet each other's ideas. And then what should we do in the future is, again, going back, sorry. I wish I could start it at the beginning, but I don't know how. So you're responding to the COVID-19 pandemic. It's important for your team to understand the methods that can be used to stop or slow the pandemic. So they need to learn how a vaccination could change the spread of the disease. So they learn a little bit about the history of vaccination. This poor little person has smallpox. They move on to thinking about mathematical modeling, and this little clip from the movie Contagion it's a pretty nice, oh, now they're not gonna let me see it right now. So it's actually from the movie Contagion, and we're gonna have to fix that now, Jen. And so what she says in there is, please hold, I need to get rid of that, is that what we need to determine is, for every person who gets sick, how many other people are they likely to infect, which is the really important calculation that you have to come up with for a contagious disease. And it's called R0, the basic reproduction number. And they're walked through how the R0 is determined, and they have a little video about what is herd immunity. And then they use this little herd immunity simulator here, where you can put the immunization rate in, and you can run a simulation of watching how the disease spreads, which is, it's a pretty neat simulation. It gives you the results based on the vaccination rate and the R0, which is this infection rate if not vaccinated here. And then we've created a spreadsheet in Google Sheets where they can enter their own data and try to see how the infection spreads. So they can change the R0, they can change the interval, that the average number of days it takes for the infected person to infect the R0 number, and the number of days from diagnosis to removal, either by recovery or death. So, and then they can just run this calculation, which is, gives some very interesting curves, and depending upon how they manipulate these numbers, they will get a lot of different curves of the infection. And then they can also look at the data over here, so that if you wanted to do a much more intricate analysis, you could use the real data here. It's a basic spreadsheet. It is certainly not completely faithful to reality, but it gives you a sense of exponential spread and exponential disease. And then again, they have to figure out which of the diseases are most contagious, and they're given that information earlier. And they have a question about Edward Jenner, and then another, and then, oh, we have 4 here, I forgot that. And then some discussion questions again. And then the final lesson is about developing a vaccine. And it's April, COVID spread all over the world. It's clear that the pandemic is still spreading. The vaccine is super important now, and your WHO team is in charge of monitoring vaccine development. They learn about the types of vaccines. There's a nice audio clip here from CNET about how scientists are developing a vaccine for COVID-19. You can read this article about the RNA vaccines from the conversation, and then another article from the New York Times about the coronavirus genome. And again, these are all assets that are linked outside, but they're super timely and very relatable to kids. And then WHO publishes their draft landscape of candidate vaccines, so they link to that here, and they can see what's in development. And then again, assessments. And your final discussion questions. So it's meant to be timely, it's meant to be engaging and interesting to kids. It's very usable in the remote learning situation. It's also usable when you're in class because the discussions would be very rich and could be assigned to kids. We think each of the individual pathways shouldn't take more than 45 minutes to an hour to go through, with the exception of the viruses one, which has a lot more reading in it, so it could take longer.
That was going to be my question. We've had a few different versions of this course or class. So we had gotten the question about how long do you think it would take? So Kristen, when we had developed a different format of this, we had said with supplemental material and working through everything, we'd said like 5 weeks, but if you just went through, it would be about 2 weeks every day, right?
Yeah, I don't think we thought it was going to take 5 weeks of 5,
No.
depending upon how your classes run, of 6 hours a week. We felt like you would want to sort of break it up into a 5-week chunk, but it wasn't going to take all of your class time for every one of those 5 weeks. We were just thinking that would be a nice way to chunk it out.
Right. But if you did it every day, well, you have the 6
If you did it every day,
2 weeks.
maybe, I mean, I'm going to go by hours. I would say it would be 8 to 9 hours of time.
So now what you can do with this on LabXchange is you can clone this pathway, the class, right? And you can clone each pathway within the course, and you can take things out or add things in if you want. If you don't like how they were set up, or you don't have enough time, or you have more time, and you can make your own class based on what we developed here.
I can't clone the class, right? Though, Jenna, I can't remember how to
You'd
do
have
it.
to clone, right? So no, that's a good question. I misspoke. You can't actually clone the class. You can clone each pathway within to make your own class.
Again, here I clone it. That
Yeah.
goes into my, I think.
Yep.
There,
Yep.
now the pathway is cloned into my assets that I have in my educator dashboard, which is loading. And then I could use it, and when you have it yourself, you can move the assets around if you want. You can add new assets in. It becomes your own text, your own class that you can do anything you want with the exception of you can't edit, or they can edit this, and it becomes their own. Is that right? Or am I only able to edit it because it was mine originally? I just can't remember, Jen.
Well, once you clone a pathway, you can edit assets if you make them, if
they're private, right? You can't clone. So I couldn't take something from Khan Academy that's a public asset and change anything on it.
Right.
But since this is a private one.
So then this would become your own version of this asset. Our version of it would remain the same in the original class, but you would have changed this. You'd have your name as the author, and you could change the image. You could add in text here. You could add in pictures. You could change the name of it.
One of the things I would recommend doing, so as you're going through, you know how you always wanted to go back to that first part, is just creating a link, like You know what I mean?
Yeah.
To that within the pathway that you're working in, so that when you finish it, you click on this link and it returns you back.
Yeah, that's a
That
good
might be
idea.
a way to get around the whole, like, how much have you completed?
Yeah, that's a good idea. I like that. We could edit it to do that.
Yeah.
And just have something under here that says click here to go back to the beginning or something like
Right.
that.
And then you just click and it would dump you in.
Good idea. Let's do that.
That's why Jen keeps me around.
Yeah. So does anybody have any questions specifically about the mystery illness pathway or course, sorry, class? My terminology is not great.
We had one question about Maribel, great question. So Maribel asked, and you can unmute yourself too, or I can read it, doesn't matter. Once the pathway's cloned, the links to the outside sources, do those remain active, including the videos and spreadsheets? Yes.
Yes.
Everything stays the same. And as you notice on one spreadsheet that Kristen was talking about, where the students can actually, there was a spreadsheet back there. Yes. So see how Kristen has it sent to view only? So they'd have to put that into their own drive, right? You'd have to copy that for them to be able to make any of the changes. So they have to make a copy to their own Google Drive, and then they can make any edits if they want.
Yeah, and this is a public resource. We created this at EDC, and you are more than welcome to clone it to your own Google Drive and use it for whatever you want to do. I mean, it's lovely if you use the course as well, but this is for you to use if you want to use it.
Any other questions?
Carol, I would say the reading level is relatively high in my opinion, and the bioinformatics tools they're not, most of the regular high school, they don't use those tools. So I would say this probably would be better for an advanced biology AP IB. But I mean, there's nothing extra special about this. So if you felt like you could get your kids through it, I think it's perfectly reasonable to have regular high school level students use it.
And Carol, one of the things that's nice about creating stuff on the platform is you can assign more to an individual student. Say you have a student that, you know, is that always done first, way ahead of the rest, hungry for knowledge. You can actually assign individual pieces to them that no one else will know, if Kristen wants to throw that up in her class.
Yeah.
And also message another student and say, hey, listen, why don't you skip parts 2 and 4 and just try to make sure you complete parts 1, 3, and 5. If you need any help, let me know. And message a student individually as well. Or tell them, instead of part 2, I want you to do this one that might be a little bit easier to read, or have a better, like, you know, more images so it won't scare them as much about the reading, something like that, where you can kind of adjust, which is really helpful in the online learning platform to be able to differentiate your instruction that way.
Now that's a good idea, because like you said, if you had like this kid who's just above everybody else, and rather than having him do something that's maybe, you know, not necessary, hey, skip these parts, and, you know, we're not counting this as part of your grade, so just jump ahead, and here's something maybe a little bit more difficult that I know you can handle but isn't for everybody else in the class. That's nice.
Yeah, and you could also, for example, this one I think is the part of the course that has the most difficult readings. So the OpenStax biology is a higher level reading level. You could easily, if your class was not ready for that, you could replace this with your own version of background on viruses, and you know, offer either both or just replace it altogether. So there's a lot of ways that you can, if you have the time and energy, you can actually customize this for your own purposes. For example, this zoonosis doesn't need to be in here. You could just dump it all together.
Right.
I wouldn't, because it's really interesting, but
it is. It is.
Thank you.
anybody else have any questions? We have about 10 more minutes in this time. We're happy to also let you go, but we'd love for you to ask questions and be here for you to support you if you need anything.
Are you guys thinking about doing anything else with bioinformatics?
Yes.
Okay, that would be great.
Yeah, we're actually looking at a lot of different possibilities, and bioinformatics are A, super interesting, and B, very hot in the careers sense. So we definitely want to provide more more practice for kids using bioinformatics tools and understanding what it means to use them.
Mm-hmm.
But we always want to do it in the context of an actual problem. So instead of just having them practice with a tool, we will be building a storyline around it so that they can understand why you would use them.
Right. Now that's super. I think that's something that lends itself well to this individual virtual learning, because when you have kids in a computer lab and everybody's on the same thing at the same time, it's really hard for them to kind of get through. But if they do it on their own.
So we just had a request to clone, so cloning the cluster, which you talked about, which you actually can't clone the cluster because they're so in depth, but you can clone each individual pathway within the cluster, and then that will go into your library. So Kristen, do you mind navigating to the ABE cluster? So, okay, so you go to browse and then library. Yep, you got it.
I know how to do this.
Oh, you're going a whole different way. Oh, you're going on the page. Whoa.
Yeah, see?
I'm so set in that one way I get to it. So here's all the tours and here's the cluster. So let's say if we go pick a tour. Kristen, anyone you want to do?
I don't know why it said that to me. Here's the tools and techniques in biotechnology, micropipetting. So within this, I can actually clone. No, I can't.
If you go back.
Oh, I have to go back. Sorry.
That's okay.
Oh, no, I didn't. No, I don't want to go back there. I don't want to go back there. Don't click that. Oh, okay, here it is. Right.
Yeah,
there I can clone it. If I click it just right.
It takes a second because it's copying.
If I give it the nicest possible click, it will let me have it.
So then that's all you do, and then it's going to be in your content. So then on your educator dashboard, if you navigate to dashboard, then educator dashboard, and then you navigate to content, you will see it in your there. And you can also do favorites. I don't know if we went over that part.
I did. Yeah.
Okay. Yeah. And we had a question about cancer research.
We've been thinking about a few things around cancer research and haven't finalized anything. So if you have particular suggestions, I don't know if you guys saw my cat's tail go by. That was funny. If you have particular selection ideas, we'd be very happy to hear what you are thinking, because we want to build things that are relevant and useful to the ABE teachers. So here's my educator dashboard, and my content should be in here.
Can you show how to drill down by content type? And then if you click pathway on the left, then you'll find just your pathways that you've cloned.
So here are all of the different kinds I have here, and there's my pathways. And so this is the one I cloned from the mystery illness. And then here's biotechnology tools and techniques. And then again, it's the same here. You can do what you will with it, move things around, change the text, and make it your own. It's not loading very well right now, but whatever. There it is.
So the other thing too, if once you're on the LabXchange platform, while you're waiting for ABE curriculum to maybe be developed, there are cancer research assets within the LabXchange platform already. If you're looking for, this fall, and the curriculum might not already be up there. And if you're teaching bioinformatics, I did use some of the LabXchange platform this year to help my seniors while I was actually live, because we did mitochondrial DNA, and then we took their mitochondrial DNA, library prepped it, and put it on the sequencer, and then we had to figure out how to read that data. So we did a lot of bioinformatics stuff through LabXchange, which was extremely helpful for me. So there, once you join this platform, and if you're using this platform as a combination of remote and live situation with ABE, it's gonna open up to be able to create more learning opportunities for your students in the same subject area as we go remote. And if we are remote, let's keep our fingers crossed that it's not 100%, but there are a lot of learning opportunities in there, even with the micropipetting, like just videos on it and things like that. So you can kind of add a lot to it if you need to stretch it out, per se, for a little bit. Kids.
Jen, we'd love to have the curriculum and lab kit around breast cancer causes, just to see if we could collaborate a little bit on that.
Sure. Jan has my email, and then abeinfo@edc.org too. It'd be great.
So if anybody has any other questions, feel free. If you feel like, tomorrow you come up with a question, you can absolutely email ABE-info and we will answer your questions. And if you have particular ideas for new curriculum that would be engaging and helpful to you, then we welcome those.
I just had a thought, because Deb had done a great job explaining how many assets there are on LabXchange. So Kristen, do you mind stopping share for 1 second?
I do not mind stopping share.
Yeah, okay. So I think my LabXchange is still open. Okay, should be. Okay, so if you are in the, if I'm gonna go to the mystery illness course, which all of you are in, I don't want to play around with it too much because we have people in here. But if I wanted to take this one, and I've cloned it to my own private account, I'm not working on anything that anybody's publicly using, but if I wanted to take it and I wanted to add content to it, we didn't go over that too much in depth. But if I wanted to take this pathway, and we talked about like moving it around, and we talked about changing it, you can also add in. So if we go to edit, okay, see if it's going to work. So we'll come up with the editing window. Down at the bottom, you'll see a box. I'm not sure if it's just my computer not working, or if it's just not wanting to today. Or is it that one? Okay. Okay, so if I go down to here to add content, then I can pull content from either the library, or if I had things that I had favorited, that's where I'd pull it from, or if I had anything I created. So if I created, let's say, for my lower level learners, maybe I needed new assessment questions. Those ones are way too hard that are existing in the course. So I could pull from here, and I could add, let's say it was coming soon. And I would go ahead and I could add that item in there, and then I could move that item around. It's gonna pop up, and I could put that assessment question wherever I need to put it. And then when I publish it up here, then that would go ahead and repopulate my new class. So if you have a few different sections that you're teaching, you can create different classes for each section, and you can put different assets in each section according to their learning level too. So I just wanted to point that out when we were talking. And it is almost the end of our time. Does anybody have any more questions or anything? I don't want to keep you over. I'm going to delete this before I forget. And we will fix that video that's no longer showing. We do have, just to wrap up our webinar for today, Deb, did you have anything last minute that we missed?
No, just maybe mention the mentor feature of LabXchange too. If you have a specific question for LabXchange or looking for some help with learning how to use LabXchange, there's a mentor feature in the LabXchange on the main page. and you can contact teachers that use it that are willing to become mentors, and through either email or directly through LabXchange. There's contact information on the page for that.
Okay.
So
That's great. That's great.
if
And
it's
so
more of a LabXchange question than it is an ABE question, you may want to contact somebody directly there. Sorry, I just dropped something.
No problem. And that's right. And if you don't know who to contact, you can feel free to contact us and we'll put you in touch with the right people for sure. This webinar is being recorded. We'll have the links out to you shortly. You'll be getting an email that has the link to it. They will stay active for quite a while. So Maribel, you can have it as long as you want. And I will have the permanent recording, Maribel. So if for some reason you can't find it on the website anymore, you can just contact us. With that webinar, we're also going to ask for some feedback. The link will be in that email. So if you could send us feedback, we really appreciate it. We value what you have to say. So thank you all so much for coming, and we hope to see you soon. Have a good night, everybody, or good day.
Thank you.
That time change always gets me. Bye-bye.
Bye-bye.
Good night. Thank you.